Welcome friends to a new session on genetics. In this session, we will discuss different types of traits related with sex, sex-linked inheritance, which includes eye color in Drosophila, hemophilia in man, Hollandric inheritance, sex-limited and sex-influenced characters, biological basis of heredity, sex-linked genes and their inheritance. Organisms have morphological sex difference as males and females. Generally, sex chromosomes which contain sex determining genes determine the sex. Thus, chromosomes which determine the sex of an organism are known as sex chromosomes or otherwise allosomes, and other chromosomes are called as autosomes. As sex chromosomes determines the sex of an organism, also their phenotypic expression which is restricted to a particular sex of an organism. This type of phenotypic expression and inheritance is called sex-linked inheritance, that is, certain characters are inherited to either male or female sexes and not to both. Other characters whose expression in a particular genotype depends on whether the individuals are male or female are known as sex-influenced traits. Still, another class of traits called sex-limited traits are distinguished by the fact that one form of this trait may be expressed in one sex only and not in the other. Let us see about sex-linked inheritance. Morgan's discovery of sex linkage in Drosophila melanogaster. Sex-linked inheritance is the inheritance of a trait that is determined by a gene located on one of the sex chromosomes. When parallelism was discovered between the X chromosome cycle and sex determination, it was generally assumed that genes other than sex determinants were also located on the X chromosome. The sex-linked inheritance was first discovered by Thomas Hunt Morgan. He was a Nobel Prize winner in physiology medicine in the year 1933 for discoveries elucidating the role that the chromosomes plays in heredity. In 1910, he discovered in Drosophila melanogaster this particular phenomena. A gene had undergone a change that resulted in a phenotypic alteration. This change expressed itself as white eyes, which is a mutant form, rather than the normal red eyes. The white-eyed male first discovered was crossed with a red-eyed female. All the F1 flies were red-eyed, but the F2 contain red and white-eyed flies in the ratio 3 red is to 1 white. All the white-eyed flies in the F2 were males. In the F2 generation, about half of the flies had red eyes and half had white eyes, but all the female flies had red eyes. The recessive allele was expressed only on males in this experiment. Morgan reached an explanation by associating this gene with X chromosome. Because the male fly had only one X chromosome and a Y chromosome that lacked most genes of the X. It was postulated that the allele for white eye was hemizygous. Furthermore, the mutant allele present in the X chromosome of the original white-eyed male was passed on to his daughters. All the daughters are therefore carriers of the allele. Half of the F2 males received the W plus allele and developed red eyes, and half received W allele and developed white eyes. The equal proportion of red-eyed and white-eyed F2 males was thus explained on the basis of segregation of X chromosome from the F1 mothers. Now, let us examine the salient features of sex-linked inheritance. 1. Sex-linked inheritance is the inheritance of body characters controlled by genes located on sex chromosomes. 2. The sex-linked characters are transmitted along with the sex of animals. 3. The sex-linked genes are located on X or Y or both X and Y chromosomes. 4. Most of the sex-linked genes are located on the X chromosomes and their alleles are absent from Y chromosome. 5. Most of the sex-linked characters are recessive. 6. Sex-linked characters are more common in men than in women. 7. They follow crisscross inheritance or zigzag inheritance. That is, the sex character is transmitted from father to the grandson through his daughter. Now, let us see about types of sex-linked inheritance. There are 5 types of sex-linked inheritance. They are as follows. 1. X-linked inheritance. 2. Y-linked inheritance. 3. XY-linked inheritance. 4. Completely sex-linked inheritance. And 5. Incompletely sex-linked inheritance. First, let us see about X-linked inheritance. The inheritance pattern associated with the sex linkage is so obvious that is ideal for genetic studies. Early Greek philosophers noticed that some human traits tended to skip a generation. An inherited characteristic was observed in father but not in any of his children, either female or male, and then would reappear in males of the next generation. This distinctive crisscross pattern from father through daughter to grandson, replacing the usual pattern of the F1 and F2 generations, is now interpreted as evidence of sex linkage. The explanation given for sex-linked inheritance given by Morgan's experiments in Drosophila that are associated with the genes on the X chromosome. Certain sex-linked genes are located only on X chromosomes and their alleles are absent on Y chromosome. These genes are called X-linked genes. The characters are controlled by these genes are called X-linked characters, and their inheritance is called X-linked inheritance. Example: eye color in Drosophila, hemophilia in man, etc. In order to understand the inheritance of characters present on sex chromosome, let us consider the transmission of X chromosome from male individual in man or in Drosophila. As illustrated in this figure, X chromosome from male individual will always go to daughter, while X chromosome from female individual will be distributed amongst both daughters and sons. Consequently, if female parent carries recessive character in homozygous condition and male parent carries dominant allele, female individuals in F1 generation will always show dominant phenotype, that is heterozygous condition, and male individual will always show only recessive phenotype in this generation of F1. If intercrossing now allowed among individual from F1 generation, it would be seen that inheritance with respect to sex is of a crisscross type. A character from father goes to daughter in F1 and then from daughter to his grandson in F2. Let us see the case of eye color in Drosophila. In Drosophila, red eye color is dominant over white eye color. The genes for eye color are located in the X chromosome. The Y chromosome does not carry any allele for eye color. Morgan in 1910 crossed a red-eyed pure breeding female with a white-eyed male. In the F1 generation, all the offspring are red-eyed. When the F1 is inbred, white eye appears in 1/4 of the F2 generation, as in Mendel's monohybrid experiment. Of the F2 offspring, the female population in the F1 generation will consist of 50% red-eyed and 50% white-eyed individuals. Similarly, in the female population in this generation, 50% offspring would be red-eyed and 50% is white-eyed. In this cross, red-eyed female transmits the gene for red eye color to all F1 offsprings through her X chromosome. The white-eyed male transmits his white eye color gene to his daughter through X chromosome. So, F1 offsprings are red-eyed. The female is thus heterozygous and the male is hemizygous. The hemizygous condition is a type of zygote in which only one allele of a pair is present. In a reciprocal cross, when a red male is crossed with a white female in the F1 generation, all the females are red-eyed and all the males are white-eyed and are produced in equal numbers. When these F1 offsprings are interbred in F2 generation, female individuals would be exclusively red-eyed, but in male population in F2, 50% individuals would be red-eyed and 50% white-eyed. In this cross, white-eyed female transmits white eye color gene to all her sons and daughters through the X chromosome. The red-eyed male transmits red eye color gene capital W to his daughters through X chromosomes. So, the F1 females are red and males are white-eyed. From the above reciprocal crosses, we can see that the trait located on a sex chromosome would alternate sex from one generation to next generation. In other words, the character is transferred from mother to her son and never from the father to the son. Let us see in detail about sex linkage in human beings. In human beings, there are 46 chromosomes, that is 23 pairs, present in each somatic cell. In female individuals, there are 22 pairs of autosomes and 1 pair of X chromosomes. And in male individuals, there are 22 pairs of autosomes and 1 X and 1 Y chromosome. Since female will produce one type of gametes, gametes from male individual will determine the sex of the progeny. Sex-linked characters in human beings will therefore follow the same pattern in Drosophila. Hemophilia is one of the important examples of sex-linked character in human beings. Let us see about hemophilia in man. It is a hereditary blood disease discovered by John Conrad Cotto in 1803. It is a recessive character and is therefore masked in the heterozygous condition. Individuals suffering with this disease lack a protein factor responsible for blood clotting. Consequently, even a minor cut may cause prolonged bleeding leading to death. This is because of the absence of a factor in the blood called anti-hemophilic globulin, which plays an important role in blood clotting. In normal persons, the blood clots in 2 to 8 minutes, but in hemophilic patients, clotting is delayed for 20 minutes to 24 hours. Hence, they bleed continuously from the wound. So, hemophilia is also called bleeder's disease. Since it is a recessive character, a female may carry the disease and would transmit the disease to 50% of her sons, even if the father is normal. It is caused by recessive genes represented by small h, and the normal condition is due to the dominant gene capital H. As other sex-linked characters, it is common in men but rare in women. Generally, hemophilic patients will die before reaching reproductive stage if they are exposed to severe bleeding. Hemophilia is appeared as a mutant in Queen Victoria, and from her it was inherited to her descendants. Hence, this disease is common among the royal family of Queen Victoria. So, this disease is also called a royal disease. Hemophilia, the bleeder's disease, follows criss-cross inheritance. It is transmitted from the father to grandson through his daughter. Let us see about carrier mother genetics. There are 4 possible outcomes when a carrier mother and a normal father create a child for each pregnancy. As illustrated in this figure, they can have a daughter not a carrier, a son without hemophilia, a son with hemophilia, and a daughter who is a carrier. Whether the daughters are carriers or the sons have hemophilia depend upon which X chromosome they receive from mother. Let us examine the case where the father is with the hemophilia. There are 2 possible outcomes when a hemophilic father and a normal mother create a child for each pregnancy. The daughters will always receive an unaffected X chromosome from the mother and affected X chromosome for the hemophilia from father, resulting in daughters all are carriers. The sons will always receive an unaffected X chromosome from mother and an unaffected Y chromosome from the father, resulting in sons all without hemophilia. As illustrated in this figure, all of the daughters will be carriers and none of his sons will be affected by hemophilia. Let us examine the case of father with hemophilia and mother as carrier, that is criss-cross or zigzag inheritance. Genes present on X chromosomes show crisscross inheritance, and this is illustrated in this figure. During reproduction, when a male has a defective sex-linked gene located on X chromosome, he transmits this defective X chromosome only to his daughter, while the female carrying this gene transmits it to her daughters and son both in equal probability. So, the male individual passes on his recessive sex-linked trait to 50% of his grandsons through daughter. The sex-linked trait, being recessive, will not be expressed in females but will be expressed in males. Therefore, males suffer from a genetic defect while females are only the carriers. Females express the trait only when they are homozygous for the recessive gene. This type of inheritance sex-linked trait from father to daughter and then from daughter to her sons is known as crisscross inheritance or sex-linked X-linked inheritance. Let us examine about Hollandric inheritance or Y-linked inheritance. Hollandric inheritance or Y-linked inheritance is the determination of a phenotypic trait by an allele on the Y chromosome. The sex-linked genes located on Y chromosome only are called Y-linked genes. These genes are confined to male only, hence they are called Hollandric genes. Holos means whole, andros means male. Hollandric genes are transmitted directly from the father to the son. Their mode of inheritance is called Y-linked inheritance. Few traits are Y-linked, and the Y-linked diseases are rare because the Y chromosome is small and does not contain many genes. Since the humans who have a Y chromosome are males, Y-linked characters are passed from father to son only with no interchromosomal genetic recombination. Y chromosome deletions are a frequent genetic cause of male infertility. Molecular genetic techniques have permitted scientists to isolate individual genes on the short arm of Y chromosome and characterize them. By using these techniques, geneticists have shown that the human Y chromosome contains at least two genes, one HY and two TDF, testis determining factor, which appears to be an important sex determining gene in humans. Thus, the Y chromosome of mammals including humans contain very few genes, but they have major effects on phenotype. Examples of Y-linked inheritance are hypertrichosis, ichthyosis hystrix, etc. Let us examine about hypertrichosis. It is also called as Ambras syndrome. It is characterized by the presence of long, often dark hairs growing from the helix of the pinna or the external rim, as shown in this figure. It is a well described entity in man. It is a genetic disease that results from mutations on an autosome and can be either autosomal dominant or autosomal recessive. Autosomal hypertrichosis is dominantly inherited. Mutations on chromosome 8 have been identified with this type of hypertrichosis. Now, let us see about ichthyosis hystrix. It is a group of rare skin disorders characterized by the presence of scales that may appear anywhere on the body, ranging from mild to severe, as seen in this figure. Ichthyosis hystrix is transmitted as an autosomal dominant trait. The disease is caused by a single abnormal gene on one of the autosomal chromosomes from either parent. In this mode of inheritance, one of the parents will have the disease and that person is called the carrier. In order to inherit the disease for the child, only one parent must be the carrier. The cause of ichthyosis hystrix is a defect in the development of the precursor, that is tonofilaments, which is a structural protein. The defect is due to a mutation in the gene that encodes for these proteins. This faulty gene has been mapped to chromosome 12q13. Let us summarize the foregoing discussions on sex-linked inheritance. Organisms have morphological sex difference as males and females. Generally, sex chromosomes which contain sex determining genes determine the sex. As sex chromosomes determines the sex of an organism, the genes found on sex chromosome as linkage unit also their phenotypic expression, which is restricted to a particular sex of an organism. This type of inheritance through only one or the other combinations of sex chromosomes is called sex-linked inheritance. That is, certain characters are inherited to either male or female sexes and not to both. The sex-linked inheritance was first discovered by Thomas Hunt Morgan in 1910 in Drosophila melanogaster. There are 5 types of sex-linked inheritance. They are X-linked inheritance, Y-linked inheritance, XY-linked inheritance, completely sex-linked inheritance, and incompletely sex-linked inheritance. Certain sex-linked genes are located only on X chromosomes and their alleles are absent from Y chromosome. These genes are called X-linked genes. The characters are controlled by these genes are called X-linked characters, and the inheritance is called X-linked inheritance. Example: eye color in Drosophila, hemophilia in man, etc. Hemophilia, which is otherwise called as bleeder's disease, follows a criss-cross inheritance. It is transmitted from the father to grandson through his daughter. Y-linked inheritance, which is also called as Hollandric inheritance, is the determination of a phenotypic trait by an allele on the Y chromosome. Other characters whose expression in a particular genotype depends on whether the individuals are male or female are known as sex-influenced traits. Now, I will give you certain assignments based on the discussions we had. First, describe Hollandric inheritance or Y-linked inheritance. 2. What do you understand by sex-linked inheritance? 3. Describe the inheritance pattern of hypertrichosis. 4. Write short notes on criss-cross inheritance or zigzag inheritance. 5. Give brief account of various phenotypic expressions in males and females related to sex-linked inheritance. I will also give you certain references for further reading. First one is a book entitled Biology, 10th edition, authored by Eldra, Charles, Diana and Linda, published in 2015. A second book is again entitled Biology, 8th edition, published in 2015, authored by Eldra, Diana and Linda. A third book is entitled An Introduction to Genetic Analysis, 7th edition, published from New York in the year 2000, and it is authored by Griffiths, Miller, Suzuki, Levantine and Gelbart. A fourth reference is entitled Chromosomes and Heredity, authored by Morgan in the year 1910, published in American Naturalist, volume 44, page numbers 449 to 496. A fifth reference is entitled Concepts of Genetics, 10th edition, authored by Klug and Cummings in the year 2011, and a sixth reference is entitled Principles of Genetics, authored by Gardner and Nestad in the year 1984, published by John Wiley, New York. I will also give you certain websites for further reference. First one, www.genetics.org. A second one, www.theagricos.com, and the third one, emedicine.medscape.com. Now, we have come to the end of this session. We will meet in another session with a new topic. Until then, bye.